So, hi everybody. My name is Chérif Matta. I'm a professor of chemistry and physics at Saint Vincent University. And welcome to the program. And today it's my great pleasure to interview and introduce Professor Tamara Franz-Odendaal, who is professor of evolutionary biology here at Saint Vincent University in Canada. And we are going to talk a little bit about Professor Franz-Odendaal's research. And this research has been recently awarded by several accolades, recently and in the past, actually. Very recently, Professor Franz-Odendaal has been elected as president of the Canadian Zoological Society. And she has also was awarded a very prestigious, she was elected for the very prestigious fellowship of the American Association for the Advancement of Science. So why don't we start from the beginning, Tamara? So would you be able to tell the audience a little bit about your background? Where are you coming from? How did you end up in biology? Why are you here in Canada in particular, given that you come from South Africa?
Yeah,
Thank
sure,
you.
no problem. Thanks, Chérif. It's great to be here. So I originate from South Africa. I grew up there. I did all my university qualifications there. And then when I finished my PhD, I decided I wanted international experience. So the plan was to go somewhere outside South Africa and then maybe go back. Well, I ended up in Halifax here in Nova Scotia, and I did a postdoctoral research stint with a professor at Dalhousie University. And after that, I ended up getting a position here at the Mount in the Department of Biology, and I'm now chair of the department. And
But you used to be.
You
used to be chair of the department at some point, right?
Yes, in the past, yeah. So it's been quite a journey. I certainly, growing up in South Africa, it was very different than what it's like in Canada, and I could never have imagined that I would be here as a faculty member at a university in Canada. So that's a little bit about where I came from. Why my interest in biology in general. As a child, I was spending a lot of time outdoors. South Africa has a very warm climate, unlike in Canada, and always curious about the insects in the garden, the flowers in the garden, the animals I saw around, and I just became fascinated with watching biology. And so it was a natural fit for me to stay in a biology program. I almost went into a chemistry program, but I stayed on the biology side of things. And when I finished my undergrad degree, I was looking at what I would do next. And at that point, I decided I would do an honors program, which is a one-year research program, and actually did not get accepted into my first choice and ended up doing it in the anatomy and cell biology department at the medical school. At the University of Cape Town in South Africa. And that just opened my eyes into a whole another world of the medical side of biology, which I hadn't been exposed to prior to that. And so I stayed and did a master's degree in a field called developmental biology. And then after my master's, I wasn't quite sure what to do, which I think is very natural. Trying to figure out what you want to do in life. And so I worked actually in a human genetics lab doing diagnostics of human disease.
And where is that?
Also at the University of Cape Town, on the next floor in the same building where I did my master's. And after that, I decided, no, I really needed to go overseas. So I went to the UK and I worked in business actually for 2 years in a pharmaceutical company. And decided that was not for me.
Were you in research and development, or were you in the business side of things?
I was monitoring drug regulations for Eastern Europe. So it was in the corporate business company, a big pharmaceutical company. But it was a little bit related to science because I was reading the drug regulations. So it was great, but I discovered that I really liked the research side, the discovery side of science, and I really liked biology. And so I thought, okay, I really have to do this PhD that I was contemplating for so long. And then I couldn't figure out what I should do my PhD in.
And that was where?
I was currently in the UK at that point, in London. And I couldn't figure out what to do my PhD in. But I was kind of feeling a little homesick, and I felt I had to go back to South Africa. So I chose a very South African PhD, and I was studying fossils from South Africa. So I switched gears from doing kind of developmental biology and anatomy side of things to fossils and geology and paleontology, which was super cool.
I was going to ask you, what exactly is paleontology? I read this in your background, and I don't want to interrupt you, but if you can.
Yeah, that's fine. Yeah. So paleontology is the study of animals that lived in the past. They lived like 2 million years and older than that.
Right.
So fossilized remains of animals tend to be the skeleton that fossilizes. You can see where this is heading, Chérif. And when I finished my PhD, I was like, okay, well, I should do a postdoc. And I trying to figure out what to do. And I discovered this lab at Dalhousie University, who's somebody who was one of the pioneers in this field called evolutionary developmental biology, which is a way of looking at the evolution of animals, but through the lens of a developmental biologist. So you see a fossilized animal, and it's got this gigantic skull or these huge teeth, and then you ask the question, well, how did that actually develop? Like, what is the genetic program that enabled the skull to get that big? And that's kind of this merge of developmental biology and evolutionary or paleontology. And so this person at Dalhousie, Dr. Brian Hall, I reached out to him and I said, hey, I'm in South Africa. Can I come and do a postdoc with you? And he said yes, and I was delighted. And so my husband and I came to Halifax to do my postdoc. And so I started working in this field of evolutionary developmental biology.
And the rest is history.
And the rest is history.
By the way, we share the same continent because my roots are from Egypt, North Africa.
Yep.
So we are both from the same continent. Exactly,
yeah. And my mother was born in Egypt. So,
I remember you telling me this one time. Yeah. So I understand that your research touches on both the fundamental and the applied side of things. And that's something I have also a great interest in, especially since my postdoctoral time when my former supervisor, Professor John Polanyi, used to say, and I'm not sure if this is a quotation he was making or is this an original statement, but he said something to the effect of the following. He said, in order to have applied science, you have first to have science to apply. Now, I don't know if this is John's statement or he was quoting someone else. So I leave it to you. What exactly are the two branches of your research, Tamara, and how they interact, and what do you see the connection between the fundamental and the applied?
Yeah, that's a great question. So fundamental science, let me start off with saying what the difference is between fundamental science and applied science. So I think most people in the general public are familiar with applied science, and that is you're doing a research question, you're trying to solve a very specific problem. So you're trying to cure this disease, and you're trying to figure out, well, how does this disease manifest in the human body? What's controlling that disease? So the science behind that is going to be applied in a very clear, within the next 5-year time frame, you know how it's going to be used. Fundamental science is figuring out answers to fundamental biological questions. So how does this cell communicate with this cell without any relation, or not even thinking about a disease specifically, but just how do these two cells, for example, talk to each other? And that knowledge is important because, and it's important to the applied scientists, because when they are trying to study how to solve or cure a disease, they need to know how cells communicate. They need to know how receptors on cell membranes function. And so we need the basic cell biology. We need to understand how an embryo develops normally so we can understand what goes wrong when it doesn't develop in the typical way. So that's the difference between applied and fundamental science. In my research group, we're doing both applied and fundamental science, and we have two two main arms of our research. On the fundamental side, so we're answering a question because we're curious about the answer. So on that side, we are studying...
So the fundamental is mainly driven by curiosity and understanding nature.
Yeah, understanding how
not
biology
curing a
works.
specific disease
Exactly,
or something. Yeah.
exactly. So we don't know what the application is. We know we're trying to, as you say, satisfy our own curiosity, but gain important scientific knowledge that will be used down the road. No doubt it's going to be important down the road, but I cannot predict right now how it will be used. So that's fundamental science. So in our lab on the fundamental side, we are studying a ring of bones in the eyes that is present in the eyes of many animals. And so I should backstep a little bit and say that my research group studies bone development. And the reason we study bones is because of my background in paleontology, where the bone is the thing that fossilizes, and then my curiosity and my background in developmental biology, which wants to understand how do those features appear or arise over evolution.
Can there be other type of tissues that fossilize, or it has to be bone only?
Bone and teeth are fossilized.
Bone and teeth, yeah.
And it really depends on the circumstances under which the animal died. So the bones may be preserved well or not preserved well, and that all depends on the conditions when the animal died. And so there's a lot of chemistry actually involved in that, and
mineralization of the minerals.
Yeah, so sometimes we can have some soft tissues can fossilize, but then the organism has died and it's trapped in amber, for example, or something, right? So there's something else that's helping with the preservation. But generally speaking, it's the skeleton, so the bones and the teeth. So in my lab, we're trying to understand the development of these bones in the eyes that, as I said, are present in many animals. And the question started from the paleontology side. So the question was, why do some animals that lived 100 million years ago have these bones in their eyes and other animals don't? Well, if I want to answer that question, I have to understand how these bones actually develop in the first place. So that's the one side of our research, is trying to understand, well, what is the genetic mechanism that's causing these bones to develop in the first place?
And that's the fundamental part.
And that's the fundamental side. So the curiosity there is trying to solve the evolutionary question, like why do some animals have them and some animals, and sometimes related animals. So that's a really interesting question to figure out why some animals have them. And for example, we discovered that some animals had they evolved with them, and then they actually lost these elements secondarily after they first evolved them. So that's another whole mechanism. This is a discovery
from your group.
Yeah. So that's another aspect of the fundamental research. And then on the applied side, and this is a more recent, in the last 8 years or so, we've become very much fascinated on how the environment is affecting bone development. And specifically, we're interested in how vibrations in the natural world, and as well how simulated microgravity affects the skeleton. So this research is primarily funded by
vibrations in the
yeah.
micro world and the microgravity.
Yeah. So this research is this research is primarily funded by the Canadian Space Agency. And so they're really interested in, when astronauts go into space, when they come back to Earth, there's damage to the skeleton. Mm-hmm. So how can we mitigate that? How can we make that damage less? Or, really, and we need to fully understand what happens.
But Tamara, how does vibration affect the astronauts? I mean, they are... sorry.
You're jumping ahead.
Yeah, sorry.
So
I should stay quiet, but I'm very interested in what you're saying.
This is great. Yeah. So we're trying to understand the effects of microgravity on the skeleton. So we know that when astronauts come back to Earth, there's a reduction in the bone density. So we know there's a loss of ossification or bone development. So we're trying to understand exactly which cells are involved in causing that loss. In another aspect of our research, and we have shown that if you use vibration treatments, you cause the opposite effect. So you cause extra bone to form. So now the question is, can you use vibration as a countermeasure to the microgravity? So we first have to understand which cells are involved in each of those before we can start like combining them and see if the one can mitigate the other. And this is a really interesting strategy, and it's one that we're doing in a model organism called a zebrafish. So it's a little aquatic fish that has the same bone cells as a human. So it's a great organism, and the advantage
is that why you study mainly fish? Is that or is it correct first that you study mainly fish, and is that the reason?
It's partly correct. So in our applied research, we study fish, and the main reason is because we can study it from day one of development all the way to when it's an old adult, like an adult. So we can study the entire life history of the animal. So it's a perfect organism to do these sorts of studies. In our fundamental side of our research, we actually use a different model organism, and we use chicken embryos. And we do use chicken embryos because these embryos are huge. And so we can see a lot more than what's going on because everything's at a macro scale. So you can imagine a chicken embryo fits inside an egg, so that's kind of the scale we're looking at. And if you think of a zebrafish, which is about a 2 centimeter fish, its eggs are like 0.5 millimeter. So we have, a huge difference in scale. So we choose model organisms to suit the question we're trying to answer.
Very interesting, very interesting. Now, I will ask a question that is probably not very intelligent, but that comes from what you just said. Does your research has any bearings, for example, on something like osteoporosis that many women, let's say, suffer from, especially after menopause and so on?
Yeah, so that's a great question, actually. And so very recently, vibration treatment has started to be used as a almost like a treatment for osteoporosis because it causes this extra ossification. So it's starting to be used as a treatment, but we still have to fundamentally understand what's going on at a cell level, like there's 3 main bone cell types, and we don't actually know which one is the one that's responding first to a treatment. Yeah. And so if we're going to ever, decide we're not going to use a vibration treatment, we want to use a drug, then we need to know exactly what we're targeting.
Yeah, it's very interesting because, the vibration is a macroscopical thing, but the bone, the chemistry of the bone is a molecular, atomic process. Yeah. So how does one go from the macroscopic to the microscopic? And also, I was going to say, is there truth in the fact, in what appears to be a commonly known fact that weight-bearing exercises strengthen bone density and so on? And again, what is the connection? Or is there a connection between the macroscopic lifting of weights and the biochemistry? How can this lifting of weight influences the biochemistry so that you fix more calcium, I guess, into the bones? Yeah.
So I think one thing that people tend not to think about is that the skeleton and the bone tissue is a very dynamic tissue. It's not just the scaffold that's holding your body together. It's actually super dynamic. So if you imagine you break your arm, there's repair that happens, right? And so there are cells in the bone that can sense disturbances to the bone. So, and that could be a muscle pulling on the bone in a certain way or straining it, and they sense that. And then they communicate to other cells in the bone saying either deposit more bone or resorb bone. And so there's this communication between these 3 cell types that are continuously going on. And so one of the questions we're trying to figure out with our vibration and microgravity work is which of those 3 cells is the first one that's actually responding at a genetic level.
Okay, well, thank you very much. This is great. I was going to ask you about another aspect. You are a professor, you're a university professor and a very accomplished one. And of course, as a university professor, you have other responsibilities, such as teaching, for example. Now, how do you view teaching in connection with your academic research, let's say? Is it reinforcing it? Is it taking time off from the lab for you? Or how is it affecting or not affecting your research, let's say?
Yeah, well, it's certainly different on different days and weeks of the year. So there are times when my research and teaching, you can't even see the line between them, because I have students who are doing research projects with me as part of coursework. And so they're integrated, completely integrated. And then there are other times when I'm teaching in a classroom, in a bigger classroom, and I can bring my research into whatever the topic that I'm teaching, and tell students, and I try to do this as much as possible, tell students about the research I'm doing. And, that's a way of starting to engage students in the research. Yes, certainly, by physically being in a classroom teaching, it's taking time away from me to be in the lab doing research, but it's really an integral part of engaging the curiosity of the students in the class. And
do you use the class also as a recruiting ground for your research students in the summer, or
exactly?
Honor
So
students?
by sharing some of the research we do in the classroom setting, students will then come up to me afterwards, or maybe 2 months later, and say, I remember the talk you gave about your research. Do you have any opportunities in the lab? Could I do a research project? Could I volunteer? Could I work? Could I have a summer project? And so that's how it's an integral part of building the research lab as well.
How do you pick your research students? Like, what is the first thing that you look at? Like, this person I want in my lab, this one, I'm not so sure.
Yeah. That's a tough question. I think for me, it comes down to how enthusiastic and passionate they are about the subject matter. Fairly often, students who are, the top scorers in the class are not the best practically in the lab. And so it's not automatic. I definitely don't go to, and only pick the students with the highest grades, because they're actually not the best in the lab. And there's another group of students, and they're harder to find, the ones who actually have the technical ability to work in the lab and who thoroughly enjoy that environment. And so often when a student comes to speak to me, I get my own kind of sense. I introduce them to my lab group, I get their feedback, and then I speak to our lab instructors who have closer contact with students and some of the other profs in the department, and it's like, what's your impression of the student? How are they in the lab environment? And that's very insightful.
And so if I gather from what you say is that you place more emphasis on their interest and drive rather than training, because you will provide the training anyway.
Exactly.
Right?
Yeah.
So I see. Thank you. And how about the science programs here at this university? We're a small undergraduate university. Do you see the university as a place where good science is being made? I assume the answer is in the positive, but
yes,
I want you to elaborate on this, please.
Yeah.
At Mount Saint Vincent University, that is, here in Halifax, Nova Scotia, Canada.
Yeah, at the Mount, we have some phenomenal researchers doing amazing work. We're often a little bit undiscovered, maybe by the public or the general audience. But we compete on the international stage. We're publishing in the same top journals as researchers at other universities. We're getting, large grants, the same as other universities. We may be fewer numbers of individuals, fewer number of faculty in our departments, but the faculty we have are amazing. So absolutely, and I think specifically with our training at the Mount and in our science program, we put a lot of emphasis on those practical skills. So there are a large number of courses that have lab sections attached to the lecture material. So there's the lecture class you go to, and then you go to a 3-hour lab. And that lab environment is critical to training scientists. This is where students, in the lecture they learn the content, but in the lab they really figure out, do they want to stay in science? Because if they love that lab environment, then they will do really well in science. And our university at the Mount, we really put a lot of emphasis on that. And I can see a huge difference when I recruit students from other universities into my graduate students who come into my group who just do not have the same undergrad training as what our Mount students and what we give to to our Mount students in the science program.
That bring me back to the beginning of of this conversation that we had, because in the past you have received a lot of major accolades in your career, which speaks about the quality of the science that is being made here, which is excellent. So can you tell us a little bit of the history of what kind of accolades have you received in your career? You hold a very prestigious research chair before, and you know, so maybe you can elaborate on that.
Yeah, sure.
A little bit.
So I started at the Mount in 2006, and within a couple of years, in 2011, I was awarded a national chair for women in science and engineering program, a position, and there are 5 of these chairs across the country in Canada. And
in all disciplines.
In all disciplines, so all science and engineering disciplines, there were just
only
5 women that were appointed.
In the whole country, there is. Let me get this straight.
Yeah.
It's five positions in the whole country of Canada in the science and engineering.
Yes.
Yeah.
And so each one of the 5 have a region that they serve, and they try, and I'll tell you about the objectives in a minute. But there were 5. The country was divided into 5 regions. So I was the Atlantic chair. This is funded by NSERC, the Natural Science and Engineering Research Council of Canada, that funds a large portion of my research lab as well. And so to be eligible for one of these chairs, you had to already be funded by NSERC, by the Natural Science and Engineering Research Council of Canada. You had to be a role model for women in science. So I was awarded this chair in 2011. I actually just finished in December 2023, so it was renewed once and extended a couple of times. So I held it for a large period of time. And the goals of that program it was a very large scope. And so the goals were to encourage more young girls to take science, try and help those that have selected science stay in science or engineering or computer science, help professional women who are in those fields stay in their careers. So it was a lot of professional development and outreach work. And so as part of that work, I ran regional Atlantic-wide conferences for women in STEM, science, technology, engineering, and math. And I ran a number of outreach events for students. I had a wonderful team that I was working with, that I recruited. And so we ran, one of the key things we ran was a zebrafish summer camp, which we ran every year for girls in junior high to spend 1 week on campus doing science all day long. And that we did that for almost 10 years. And so that was the NSERC chair position. So I just finished that position, as I said, in December 2023. During that time, I also became a fellow of the American Association of Anatomy, which is an international international society with 3,000 members or more across the whole world, and they have they award the status of a fellow to the society to a very small subset of their members.
And for the non-scientific audience, I will just clarify one little point here, that the status of fellow in these learned society is a very prestigious, and usually by election of the membership. It is the highest ranking, or is the highest grade of membership in these societies. And it is based on, it is usually a very small percentage of the membership that are eventually elected as fellows on the basis of their excellence in science.
So I was awarded that a few years ago. I received a number of other awards, like the Research Excellence Award at Mount Saint Vincent University, the Leadership and Equity Award at Mount Saint Vincent University. I've been the president of the Nova Scotia Institute of Science and actively engaged in that organization for probably at least 10 years. And then most recently just become the president of the Canadian Society of Zoology, and also received or was recognized as a fellow of the American Association for the Advancement of Science.
And you had an important role, Tamara, in increasing the visibility of minorities and women in science. Can you tell us a little bit about what you have done in this domain, please?
Yeah, so a lot of that work started when I was awarded the chair position in 2011, the NSERC Chair for Women in Science and Engineering, and I really got engaged in not just, providing workshops for professional women in STEM or doing outreach to girls in junior high and high school. But I really got involved in kind of the social science side of research to try and understand, well, what are the experiences of minorities in science in Canada? So what are the experiences of female postdocs?
And the roadblock, the roadblocks that these minorities face, right?
Exactly. So what are the experiences right now? Because a lot of the data is data we're getting from the US. And so it was important to get Canadian data. So what are the barriers Canadian postdocs are facing? What are the barriers Black scientists are facing? And so we know many of those barriers we know, but it's important to just to do the research and make sure, like, are they new barriers? Are the barriers staying the same? What are those barriers? And when really needs to have a good understanding of what those are at a deep level in order to be able to remove the barriers or put supports in place that are going to make the barriers reduced for that population and hopefully completely eliminated. I mean, that's the goal, is to not have any barriers.
To make science equally accessible to any person, despite of their gender, despite of their color, skin, despite of...
Yeah. So I've been very active in the Canadian Black Scientists Network. I'm one of the co-founders, actually, and their lead ally on that network. And that's been amazing, working with those phenomenal Black scientists we have in Canada. Truly amazing individuals who are doing rock star science, like incredible. And yet, before the network was established, I probably knew, I could count them on one hand how many Black scientists I knew. And I think they face a barrier that's unique to them, and it's really important for those of us who are allies to really reflect on the privilege that we have and the tables that we're at when there are discussions happening and there's not a diversity of people around the table. Like, who are we missing from this conversation? And what can we do to ensure that their voice is heard and that we can help?
Yeah, thank you. And to close this this very exciting discussion with you, where are you envisioning to take your intellectual work in the future? Where are you headed in the short term and in the long term? What is your big goal?
That's a tough question. So, I mean, it's hard because, and I'll just go on a little tangent here, but your question reminds me of, when students come to me and they say, well, I would like to work with you in your lab in like 6 months' time. And then and I said, that's great. Okay, I've got a spot for you. And then they go, what will I be working on? And I'm like, I can't tell you exactly, because the research we're going to do between now and then is going to indicate what we're going to do next. So it's very hard to say, like, this is what we're going to do, because I don't know what we're going to discover between now and then.
Great point. If you knew it, you wouldn't do the research, right?
Exactly. Yeah. But I will say, in our applied research, we really want to understand the communication between bone cells when they are experiencing something that's affecting the bone tissue itself. So understanding that communication network between those three cell types. And on the fundamental side, we are honing in on a network of a couple, like 3 or 4 genes that we're trying to figure out which are the players which are the critical players that are the key signaling molecules that are important to make these bones form. Because if we can get that answer, we can understand the question we're trying to answer, which is, why do some animals have these bones and others don't?
Wow. Dr. Tamara Franz-Odendaal, thank you for granting us this interview. We're really happy to have you.
Thank you for having me.
Thank you.
Thank you, Chérif.
Thank you. Well, that concludes our interview with Professor Tamara Franz-Odendaal. See you next time, and thank you for your attention.